Hello everybody, welcome back to the Mycology course. This is practical course, lecture 8. So this is Cihan University, Department of Medical Laboratory, fifth semester in the Medical Laboratory Department. I am Dr. Marwan Hussein and I'm going to present the practical session, lecture 8 for you. Today is the germitube. Germitube in Candida albicans. So, as you know, we talked about the Candida albicans, and we said that germitube is one of the virulence factor of Candida albicans. If you remember in the lecture, in the theory and practical as well, we previously have this about germitube. So this is one of the virulence factor, and today we're going to do the test of detecting the germitube in Candida albicans. So as a principle and definition of germitube, that the screen test to diagnostic test or to differentiate between Candida albicans from the other yeast, or I mean the Candida which are positive in germitube from the Candida from the negative germitube. So the principle is when we put the or inoculate Candida albicans in the serum at 37 centigrade in the incubation for 1 to 2 hours, and then after that we're going to do detection under microscope. You will find the filamentous, the germitube. So it's better to use rabbit or sheep serum. So serum you can isolate from the blood after you do centrifugation. After you do centrifugation, and then you are going to isolate serum from the blood. So we need serum. Today we're going to use a human serum, and it need time. It needs time. But we did. So we reculture the Candida, and we inoculate the Candida in the serum because it needs 2 hours at least. 2 to 3 hours, so 1 to 2 hours. So we did that. So today, and now it is in the incubator. This is the principle of the germitube. So detection the germitube in serum after incubation in the at 37, and it is positive in Candida albicans and Candida duplinensis. Just those, and both of the species, but the other species are negative. Why the germitube? Why Candida albicans make or form germitube? So it is associated with the increasing synthesis of protein and ribonucleic acid. So this is the answer of why they form germitube. It's associated with the increasing synthesis of protein and ribonucleic acid. So as we said, it is one of the virulence factor, and this test it is a rapid test for Candida albicans identification. As you see, this is Candida albicans SDA, and you previously have it, and you work in your hand. So this is a germitube. You will find under microscope like this. So the yeast do budding from the mother cell, and the daughter cell will bud the small pod from the yeast cell, and then will elongate to make germ a tube. The result of germitube, as we said and we talked about it, after incubation, under microscope, you will have a drop of the serum, and then on the clean slide and then cover slide, you will find the short hyphal filamentous arising from the yeast cell. with no construction at the point of the origin, of course. In dimensions of germ tube, it's it's about 3 to 4 times the length of the yeast cell. 3 to 4 times the length of the yeast cell. Yeah. Of course, with no presence of nucleus. As I said, it is positive in Candida albicans and Candida dublinensis, and the other species of Candida are negative. So this is that we can detect and differentiate between Candida albicans, which is positive, and the other, like Tropicalis, glabrata, are negative. As you see, this is germ tube under microscope. You will find like this, and negative, we talked about it. The Tropicalis and glabrata are negative, so no formation of hyphal or filamentous, no formation of germ tube in those species. Just Candida albicans and Candida dublinensis. Again, this is in the same slide. You find the positive and negative. Look, this is the positive, and the others are negative. Again, under microscope, you will find like this. Under microscope, more slides. Of course, it's Candida albicans and Candida Candida dublinensis. As I said, you know that it's pseudohyphae. So this is will make pseudohyphae, starting from germ tube, and then finally you will find, they form pseudohyphae. What about the procedure? So procedure, as you see, that it's very easy, because we previously did the reculture of Candida albicans and inoculation the Candida in the serum. So and we put in the incubator. So now today you're going to have 1 or 2 drop. One drop is enough from the serum after incubation, and then put on the slide, and then put the cover slide, and detect and examine under microscope. So you will find germ tube. I think we did 2 type or 2 species. One, Candida albicans, which is positive, and the others are negative. The other species, like Tropicalis, glabrata, or crozei. This is about a procedure. Of course, it's very easy. So I'm going to show you as well. I'm going to show you the video. You will find the video and the sample procedure, and then under microscope how it will appear. This is about our session, our test, and thank you for your listening. If you have any question, you can ask, of course. And in the lab, I will be with you, and any question or any queries you can ask. Feel free to ask everything. Thank you.